Aberrant chromatin is the starting point of many diseases. It is best characterized in cancers, making it a high-value target for oncology.
Every cell in your body has the same DNA sequence. So how is it that a brain cell is a brain cell and a skin cell is a skin cell? It's how the DNA is packaged. Certain genes are packaged such that they're inaccessible and genes are turned off, and other genes are open so they're turned on. Just like DNA packaging is important for normal cell identity, it's when that DNA packaging goes awry that you get disease. Conventionally, a lot of people have tried to go after DNA packaging by targeting single proteins or interfaces between two proteins. But what we know is that in disease, a lot of times it's the chromatin network or the DNA packaging network that really is problematic. I'm Laura, the CEO and co-founder of TippingPoint Biosciences. My co-founder, Dr. Geeta Narlikar, who's a professor at UCSF, is a world leader in chromatin biology. With our expertise, we have invented a new type of screening platform. We can identify molecules that specifically modulate the disease chromatin network without affecting the healthy ones in a test tube. Our platform is working. We are identifying an entirely new class of molecules that work in a different way. And our initial hits are showing efficacy in patient cells without being toxic. We're taking a really different approach at how we're doing drug discovery. I want you to take out all the assumptions for targeted-based drug discovery. Because we know that it's chromatin networks that are really causing the disease, we need to redefine what the target is. And the target is a chromatin network. How do you go about drugging this chromatin network? You have to create an assay that enables you to really understand how these interactions are changing. We can recapitulate a network that is known in disease, and then we can find small molecules that modulate these interactions and break only the bad and not the good interactions. The first disease indication that we're focusing on is a pediatric brainstem tumor called diffuse intrinsic pontine glioblastoma, or DIPG, because it's the best proof of concept for our platform. In the last 4 months, we have scaled this assay so that we can screen for 10,000 molecules now. We know that there are many diseases driven by aberrant chromatin networks. DIPG is just the tip of the iceberg, and we are planning to go after many other diseases, including other cancers, neurodegenerative diseases, and also developmental disorders. Biology is complex, and reducing it to single interactions or interfaces has only taken it so far. At Tipping Point, we are not just building a new path to the same destination, we're unlocking a new destination entirely.